Hello, what I'm going to do right now is talk about the chemicals of life for biochemistry. Start with CHNOPS. CHNOPS is a way to actually remember the elements that are actually important in terms of life. This is a picture that I found a few years ago that actually shows pretty well what's going on. It shows you the hydrogen and carbon comes together to make what are called hydrocarbon compounds. These are compounds used by life, not necessarily always coming from life, because we found these in materials coming from space. Throw in a little oxygen and you can get carbohydrates and lipids. That is truly a life form. If you can find carbohydrates and lipids, you've probably found something that came from life. Throw in nitrogen, you can get amino acids, which is one of the four chemicals, the Fab Four, and proteins. Throw a little sulfur in there, you can get a wider range of amino acids and proteins, and then throw some phosphorus in there, potassium in there, and you can actually get, for potassium compounds, you get nucleic acids, RNA, and DNA. This is a picture of kids that I had, I think this is a couple years ago. This is actual DNA material that we're going to talk about because that's going to be one of the four chemical and the nucleic acids down here. This is actually coming from strawberry. And the DNA actually sits inside the nucleus. So in order to get that, we have to break down this cell membrane, which are made out of lipids. And we have to break down the other membrane around the nucleus so we can actually get those out. And then we use ice cold, and we use detergents for that. And the same reason we use detergents to clean things. It does a really good job of breaking down material. And then we're going to use ice cold alcohol to actually get the DNA to precipitate out. And then we'll use a plastic stir rod to kind of swirl it up like cotton candy, and then you can pull it out of the container, although I think it actually looks cooler when it's submerged. But we'll do that when we actually get into DNA, so not right now. Okay, this is something that I stole from another biology teacher, actually who teaches out in Bozeman, Montana. And these are going to be the biological molecules. This level right here, the biological molecules. These are also what I call the Fab Four. Why can't I draw that? The Fab Four. Boy, this is being a pain. Don't like those at all. It's almost like I'm doing this with my tablet in class. This tablet's much better. But anyway, we have carbohydrates. That's a biological material. We have lipids. We have proteins. And we have nucleic acids. These guys are like the cinder block wall in the classroom. They're actually made out of bricks and mortar and posters and pictures and hangings and glow-in-the-dark things. Each one of those are what are called monomers, like the one cinder block or the mortar or the posters. And taking these monomers, we can actually make different things. Carbohydrates, this is the monomer row. And you can see, well, actually carbohydrates, these are like sugars and starches and some other things that we'll use for actually building and structure. They're actually made from sugar. And the difference between them is how they're actually bonded together. If they're single solitary things, it's glucose. That's the C6H12O6, and I do want you to understand, know, and be able to recite that anytime I ask. This is the basic building blocks of all the carbohydrates, and we'll talk about carbohydrates first. Lipids, the second one, these are fats. And fats actually are really important in terms of like the cell membrane. It's in terms of really important in terms of high energy, and carbohydrates are high energy also, or energy. Lipids are even higher energy, and they're going to be basically differentiated in terms of the polarity, whether they're positive, negative, whether they're hydrophilic, hydrophobic, and we'll talk about all those things later too. Number three are the proteins. Proteins are important because they basically make everything that you see in me and everything I see in you. They make up bodies. They make up muscles. They make up bones. They make up skin. They make up hair. They make up eyes. They make up everything. Proteins are made out of amino acids. There are 20 important amino acids that you need to get from your diet, and those amino acids are different because of what are called the R groups, and again we'll talk about those. Nucleic acids, these are things like DNA, RNA, and they actually contain the genetic material that you will pass on, the material that you got from your parents, and they're made up of nucleotides. They're going to be differentiated also because of groups that hang off them. But if we talk about all those things, they're important in terms of energy. They're important in terms of making up basically everything that's in a body, and they're important in terms of the genetic material that's going to get passed on. Okay, how do we take these monomers and how do we stick them together to make carbohydrates? How do we actually take these amino acids for proteins, the sugar for carbohydrates, and these nucleotides? How do we get that other, it's not a different one, how do we get it to move this way? How do we get those 20 amino acids to make all the proteins? And there are more than 20 amino acids. These are the only 20 that you need, although you get more from the things you eat. And from those, you'll make all the proteins that are you. And how do we get these nucleotides to actually become the nucleic acids, the RNA, the DNA? We do that by a process called hydration synthesis. I'm going to show you a couple of things about hydration synthesis. And the first one I want to show you for dehydration synthesis will actually show you this and a process called hydrolysis. Dehydration, D meaning to take away, hydration meaning water. I think most of this slide will show up, but this is an animation that I have in a PowerPoint that I've used in years past. I'll probably show this to you in class too. But what you do is you get an enzyme that comes together and takes the 3 monomers and makes what's called a polymer. In this case, they were 3 sugars, so it is a polysaccharide. Sacchariding means the sugar. Now, that was dehydration, and if you actually take a look at that, what we did is we lost the water. This O and that OH came together to come off as water. Okay? Hydrolysis by hydrolase actually brings in the water and rips apart the material. So we put them together by a process called dehydration. Notice the water drop taking away. We put them together and we pull them apart. Okay, so that's the first thing I want to show you. The next thing I want to show you is this website. I'm hoping you can see this. If I can slide this over. I can't seem to make it any bigger. But anyway, this is 2 amino acids, and if you take a look at the amino acids, we'll talk about what forms this. But you've got an OH, a hydroxyl, and you've got a hydrogen atom all being held together. And dehydration synthesis, we're going to combine that H. Can I draw on this? Can't draw this. We're going to put those together. We're going to pull out the water, and we're going to make a peptide bond between the carbon and the nitrogen. The carbon needs to have 4 bonding points. The nitrogen needs to have 3, and if you take a look at it, we put the water back in. We have that OH, there's the 4, and we put the H there, there's the 3, so we can do that. Metabolism, this is carbohydrate metabolism. So you take in a sugar. This is a glucose molecule and a fructose, which is called sucrose. This is what we actually use in like our sugar containers. It is the sugar from sugar cane, sugar beets, the material we actually use for our sugar. You could actually use glucose and fructose, and we'll talk about the sweetness of those in a couple seconds. But dehydration, we're going to take the water out, and we're going to actually bond them together to make the sucrose. And then hydrolysis, we're going to put the water back in. Now this one doesn't show you the enzyme that comes through there. This doesn't happen by itself. You need to have enzymes, which are proteins, again, things that you've created to actually help you lyse, which means pull apart, or to fuse, put those things together. And protein metabolism, the same thing. You have a peptide bond forming when you take the water out. And then hydrolysis, you put the water back in. Okay, good. Okay, so dehydration, this fuses. In other words, puts them back in. And here's another dehydration, and if you take a look, what's missing is this H right there, and that hydroxyl, and that's the H2O that gets taken out of the picture in dehydration. And then this one would be the same. This is sugars put together. This is the proteins put together, and I showed you animations of both of those. Okay, the first of the four I want to talk about are carbohydrates. These are all carbohydrates. There's sugar in the fruit, wheat, pasta, beans. There's actual sugar, fruits, vegetables, anything that's sweet. You could also throw milk and other things in here. This is a building that architects actually put together using like 11,000 sugar cubes. Hate to get that thing out in the rain. You can't see that well, but let me see if I can move this over where it's supposed to be. And now we're back. So like 11,000 sugar cubes to make that building. Carbohydrates, the monomer is glucose. And it is C6H12. I don't know why I didn't like those H's. And then O6. There's 1, 2, 3, 4, 5, and 6. If you want to count the rest of them, there's 1, 2, 3, 4, 5, 6, and take my word for it, since you can see there's 2 for each one of those, we've got 12. This is the way you would draw it. This is the way it's sort of configured. Notice that it's three-dimensional. In terms of sugar, fructose is the sweetest, and lactose, milk sugar, along with maltose, it is the least sweet. So if you really want to make something sweet, fructose is what you want to look for. We use this one as the basis for sweetness because this is what we use for our sugar. This just happens to be almost twice as sweet. Okay, we can actually, when I said bonding is the way these things are actually going, here's actually a sugar. I have my hand on here. I shouldn't have my hand on here. There's a sugar, there's a sugar, there's a sugar, there's a sugar. Here's that carbon hydrogen on the top. And you notice starch that you see in the pastas. This one right here would be a glucose molecule. There's the monomer. This is a polymer because we actually put all those together. But if you take a look, we actually have these things all sort of like in the same direction all the way through. This is harder for our body to utilize because we have to actually use hydrolysis to break these apart. But we can break those apart and use them as the energy source for our body. Starch itself cannot be used by us. Cellulose is the same thing, but can you see the difference between starch and cellulose? Here's the sugar. And here's the sugar. And there's the sugar. And I guess I should draw it like this, because these guys are upside down. So there's the top, there's the top. Notice that they're mirror images of each other, and they're bonded right next to each other. Because of that, this is a little harder for our bodies to digest. And then glycogen, you can see it's got starch and it goes off into different directions. This one actually would go into your body and come out of your body. Your body can't break this one down because it's so complex. Also chitin, the things that actually make the outside part of insects, they actually give structure. So it's not just a sweet food thing. It can also be used for other things as well. Okay, the next one is proteins. Proteins are actually made up of well, here's an amino acid, and it has a carboxyl group, which is that part. It has an R group, which is actually going to differentiate whether it's going to be any of these 20 amino acids, and these are the magic 20. These are the 20 that you've got to get from your diet. These are the 20 your body uses to make every single protein. Literally millions of different proteins, but you make them all with these 20. If you don't get this one, there are probably some proteins you can't make, which may make you sick or worse. Okay, and then we actually have this nitrogen group as well. And notice that they all have carboxyls, notice they all have nitrogens, and they all have R's. This is a different amino acid. They call this one R1 and R2. And the way they get put together, this hydroxyl and that hydrogen, dehydration synthesis, forming a peptide bond. And then you can actually put that back in with hydrolysis and break them up into amino acids again. And that's what we do when we actually eat, and you get proteins by eating things like meat and beans. If you eat a piece of meat, you get all 20 that you're supposed to get. If you eat beans, you may only get 19 of these, and you may not get this one right here, which means you have to eat, if you're actually going to be a vegan, if you actually are vegetarian and you drink milk and eat cheese and eat eggs, you can actually get all 20 you need. But if you're vegan, you got to be really careful. You need to make sure they have a diet that includes all 20 of these. When they come together, they actually make these ribbons and these little shapes that you see here. The shape is what's important, and different amino acid configurations give you different shape. Directionality, if you take a look at these, some of these actually come together so that they're backward. You actually get the nitrogen group or the carboxyl group. You can get those things in different places, and you need different enzymes like trypsin and chymotrypsin. These are actually two different enzymes that actually work on different levels. This one actually breaks enzymes down, or excuse me, proteins down into smaller groups, more like amino acids. And this one will break large proteins down into smaller groups of protein. This is what it sort of looks like in your body. And it's these shapes that are important. And we'll talk about those when we actually get into replication, especially for RNA. The third group are the lipids. Lipids are fats. They actually make food not stick. They may even make food taste good. It's this triglyceride that, like in ice cream, that actually makes things taste good. Cholesterol, you hear lots of things about cholesterol. Your body actually makes cholesterol. You don't need to bring a cholesterol in terms of your diet. Your body will actually make that, and we'll talk about that when we get into cells. It can actually make your cell membrane act differently. Fatty acid, that's like this piece right here. This is a fatty acid. And notice it's just a bunch of carbons with hydrogens bond to it, so we actually get 4 groupings. You get a bond there, you get a bond there, you get a bond there, and you get a bond there, all the way across. And then you have a glycol group, which is, it's like this piece, except I need to actually include that. So this is the glycol group. These are the fatty acids, and this makes up the basic shape of a fat. And this is why fats, if I zoom all the way back up, fats don't really have a monomer. If they did have a monomer, you'd have to say it was the glycol and the fatty acid. And then you have phospholipids, and fats are incredibly high energy. We actually use them to make the membrane around cells, as well as organelles, and we also use it for incredibly high energy, really high energy producer. Okay, you've heard things about, and actually what if you take a look at the fats, what do they all have in common? If you can see it, it's this zigzag form, and this is where we actually put a carbon right there, and a carbon right there, and a carbon right there, and a carbon right there. And you keep putting carbons in there. And if you take a look, that's what these zigs are, and that's the carbon that you see in here. Do you notice the difference right there? As well as right there. For some reason, there's 2 bonds between the carbon, and what you get is you get what are called saturated. And what is a saturated fat? It just means saturated means it's holding all it can hold. It's holding all the hydrogen that it can hold. This is an unsaturated fat. I'm just going to say unsat. And why? Because it actually has these hydrogens broken. And because of that, it forms a double bond between these carbons. And because of that, the molecule actually bends. So every place you would see this molecule bends. Now what's the difference? Saturated fats are usually going to be liquid at room temperature. That's why you keep butter in the refrigerator. If you bring it out, it gets really soft. Unsaturated fats can stay as a solid. And we found out if we bubbled hydrogen through saturated fats, like corn oil and coconut oil, we can actually make them turn into margarine, and that's the difference between butter and margarine. We're finding out saturated fats, because they stay liquid, they travel and they can plug up your digestive system. The unsaturated fats stay solid, they stay in your digestive system, they don't really get into your blood. So we're finding out these are much better fats. These guys are a little scary, although they taste really good. Last thing are the nucleic acids. Nucleic acids are things like RNA and DNA. They actually are the cytosine, guanine, adenine, and uracil for the RNA, and they're the cytosine, guanine, adenine, and notice those three are the same in both. The only difference is the language, this last one, with your thymine in there. We'll actually talk about the DNA before we talk about the RNA. The basic nucleotide that actually makes up the monomer, again, this is the monomer. The monomer is actually made up of a 5-carbon sugar. We have one, actually count with, this is the prime one, second one, third one, fourth one, and fifth one. So if you take a look at it, it actually has a 5 end where the 5 carbon is and a 3 end where the 3 carbon is, and that actually is going to differentiate. If you take a look at this, here is the actual what's called the backbone of a DNA molecule, and you can see it right there. This is the actual nitrogenous bases that are in there. That's these guys. But what happens is this excuse me. What happens is this nucleotide actually picks up this nitrogenous base, and then different nitrogenous bases will hydrogen bond. These there really is no real connection between these. They're just sort of like magnetically, although it's not magnet, it's hydrogen bonds held together. And because of that, this thing that's sticking out, this is the phosphate group, this is the 5 end. And let's actually say this one right here is the 5 end. And because of that, if you go all the way down, there's the 3, there's the 3, there's the 3. This is the 3, and actually it's called prime. So we get the 5 prime end and the 3 prime end. This is the 5 prime end, and if you follow this one all the way down, this is the 3 prime end. And if you look at the other side, the 5 prime is down a little bit, and the 3 is now at the top. So this is a 3 prime end, and this is the 5 prime end. So this one is the 3 prime and the 5 prime. And this one only has one side. So we'll say this is the 5 prime and this is the 3 prime. RNA only comes in one strand. This one actually comes in 2. And because it comes in 2, we can actually talk about them being parallel, because they actually are parallel, but they're also called anti-parallel because they're like mirror images of each other. And I don't really like the way they put these together, but they're actually saying that we need those shapes, whether they be sort of diamond shape, triangular shape, or roundish shape. They need those to actually fit together, but again, they're not really touching. They don't touch each other. They're not in contact with each other. They form hydrogen bonds that are in there, and they're weak hydrogen bonds, so they can break fairly easily, which is really good in terms of DNA replication. Okay, so those are the 4 chemicals, and what I would like you guys to be able to do is to actually know what are the monomers for each one. What are they, and how are they used in the body? All right, thank you. Bye.